Hello everyone, my name is Shivani Rawat. This summer I was able to join the Project SUPER Summer program, and through that I had the opportunity to be able to work under Dr. Lei Yu in his neuroinflammation lab. This poster will be talking about the categorization and extraction of articles for the analysis of painful diabetic neuropathy. Painful diabetic neuropathy, in short known as PDN, is a common complication of type 1 and type 2 diabetes, which can have various painful symptoms such as burning, tingling, shooting, lancing, and more. This is a complication that leads to peripheral nerve dysfunction, and currently, there are no effective therapeutic treatments to cure PDN. And so our lab's initiative is on the focus of inflammation, which is a possible mechanism pertaining to the onset and/or development of PDN. With that, we are mainly looking at CCR2, which is a chemokine receptor type 2 that could play an important role within inflammation when it pertains to PDN. This G-coupled protein receptor, a receptor for CCL2, is shown to play a role in the activation and migration of monocytes to the areas of inflammation. We are focusing on currently two isoforms of CCR2, these being CCR2A and CCR2B. Last semester, we were able to analyze articles that mentioned whether a functional and/or structural difference was present between the two isoforms of CCR2. Our hypothesis was that the difference in the isoform structure and the variation of the isoform ratio can lead to potential effective PDN treatment. After the analysis of our literature reviews, we have found data that states that there is a difference in the structure and expression between the two isoforms, thus further reinforcing our hypothesis. Knowing that, we are now working on our paper for this project and have obtained articles from our master list that we think are valuable to our paper. We also used the Google search engine to search up on a couple of diagrams that would mention a lot of the pathways that we were incorporating into our paper. Some pathways include NF-kappa-B, TNF-alpha, and possibly any other diagrams that would show us mechanisms and their connections to PDN. Because this summer we mainly focused on our paper, we did not conduct any in-person labs, and so we didn't find any statistical findings or results. However, we did decide to create our own diagrams from our extracted notes from the articles. For example, Figure 1 and Figure 2 shown are diagrams that I created from my notes that I had gathered out of the two of the selected articles. Figure 1 shows the results that one article found when they were trying to identify the role of NRF2 in Schwann cell-mediated functional recovery after sciatic nerve injury. My diagram shows what happens under hyperglycemic conditions and how those conditions then activate the TLR and NF-kappa-B pathways, which then promote inflammatory factors. It also shows when NRF2 is overexpressed, it causes an inhibition to these activated pathways, which in turn promotes Schwann cell survival. Figure 2 is yet another diagram that I created from my notes off of another article. This article focused on monocycline, which is an antibiotic that is used to treat bacterial infections. They tested monocycline on STZ rats to see if it would decrease allodynia and hyperaglesia, and whether it can decrease oxidative stress and inflammation in the spinal cord. Looking at my diagram, you can see how it reduces allodynia and hyperaglesia, and is also shown to reduce oxidation of lipids and proteins. As a newer member of this lab, I got to better understand these pathways and mechanisms and their connections to inflammation and PDN by the process of making these diagrams. So our future plans are to continue to analyze, and extract information from the rest of our selected articles. We are also going to be starting to work in the labs at Rutgers this semester. I would like to thank Dr. Lei Yu and the members of the Neuroinflammation lab for letting me be a part of this lab this summer. I have learned a lot over the past few months from my spring semester to now, and I am very grateful for the research experience and skills that I have obtained. I would also like to thank the directors and coordinators for the Project SUPER Summer program for giving me the opportunity to be able to partake in these research labs over the summer. Thank you.